Hello, welcome to the Gene Technology Lecture Video number 3. Today we're going to talk about genetic engineering and what it entails. So let's begin. Well, humans have figured out a way to manipulate DNA so that we can make new substances, basically. What does that mean? It's kind of complicated. So scientists have figured out, hey, if we change the DNA of an organism, we can engineer it, we can create things that do things differently. So we are now, you've probably heard of the term GMO, like genetically modified organisms is what it stands for, or GMO food that has been genetically modified. So scientists have figured out ways to engineer our food and things in society that we use. Basically, because DNA from all organisms is composed of nucleotides, we learned this in our last unit. We know that we figured out a way where you can cut out DNA from one organism and insert that DNA into another organism. Now, this is the technical way of genetic engineering, what you would be thinking about, actually modifying and changing DNA. Just so you know, farmers have been practicing different methods of engineering foods. For example, they've been crossbreeding. This picture is showing you a wild mustard plant. This wild mustard plant basically has been crossbred, which means we breed different plants with other plants. And this particular ancestral plant has been used to make kale, cauliflower, broccoli, cabbage, and so on and so forth. So genetic engineering in the realm of plants, scientists have been crossbreeding plants and producing new things. Like you probably noticed, like, for example, at the grocery store, I noticed this thing called a grapple, a grape and an apple that have somehow created a hybrid plant, all right? It's not very complicated to do it, believe it or not. And farmers and people in that type of work have figured out ways of doing this. Why is genetic engineering beneficial? Well, we can now create organisms that can produce things for us, like insulin, which is a medicine for diabetics, human growth hormone, which is another, some people need growth hormone for medicinal reasons. We've produced crops that are insect resistant, crops that use less fertilizer, food that is more nutritious, food that won't get frostbitten. So we've modified a lot of different things that we need to so that we can produce them a little bit easier. I believe salmon have been modified. Food salmon that we like to eat have been modified, and they're on the market. What did they do? They somehow modified a salmon that is a cross between, I believe, 3 different species. One of the species produces, larger fish, so you can have more meat. One of them species happens to grow quicker than the other one, and so on and so forth. So they've crossed them all together to make these engineered type of products. Now, recombinant DNA. This is DNA that has been recombined, which means DNA from DNA that is now composed of DNA from more than one organism. So more than one organism's DNA combined is recombinant DNA. What's this picture showing you? Firefly DNA was inserted into a tobacco plant. Now we have a tobacco plant that glows. Okay, let's go back here a second ago. These guys over here, they inserted the glow in the dark, the glow gene from jellyfish. They inserted that into these rats. Some of the rats now glow. Okay, what's this picture showing you down here? It's showing you how we insert genes into plants. A lot of times we have to infect them and do different things. So you're putting it into plants, and now these plants are expressing the gene that was inserted in them. So this is genetic engineering. Now, what is this picture showing you? Well, a lot of times when you genetically engineer an animal, for example, in this case, we're going to be genetic engineering, in this case, a goat or a sheep or something of that nature. What you're going to do is you generally do the genetic engineering in the egg. And then that egg is inserted into a host. The host gives birth now to a genetically modified organism. So the host, the parent, is not genetically modified, but the egg that we inserted in them was genetically modified, and then you produce an offspring that is now genetically modified. And in this picture, we call this offspring transgenic. Why? This organism is made from DNA from more than one source. So now it has transgenes. So DNA that came from another species, basically. Vectors. You're going to hear this term sometimes this unit. We're going to be talking about plasmids, and plasmids are vectors. Vectors is DNA that generally can carry foreign DNA. When DNA carries foreign DNA, it can insert it somewhere else. A plasmid is circular DNA that is found in bacteria, and bacteria can carry foreign DNA. So look at this picture. This is the bacterium, this is the chromosome of the bacterium, and sometimes bacteria have a separate circular piece called a plasmid. So usually much smaller than their typical chromosome. This plasmid can serve as what's a vector, which means it can carry foreign DNA. So what happens is you take DNA fragments from, for example, this could be human DNA. You insert it into the circular plasmid. That plasmid gets inserted back into a bacteria, and now when the bacteria reproduce, they're going to be making more of this recombined DNA. Now, to take this plasmid out of bacteria, it's very easy. All you got to do is put bacteria in a little vial, put it on ice, get it very cold, so that the pores in the membrane open up, and then that plasmid will slip or slide right out. How do you put it back in? You do the same thing. You put it back on ice, creates larger pores, and it can slide right back in through those membrane channel proteins, basically. Now, here's another picture showing you the process. You start with a bacterial circular plasmid. You got to cut the plasmid open, okay? You got to cut it open in one spot. Why? Because you're trying to insert this blue DNA into that one area. So you got to cut it open. So one spot cut on the plasmid, you got to cut twice. You got to cut around the blue. So 2 cuts here on the human DNA. And now you've released that blue piece, that blue gene that you now want to insert into the plasmid. Now, if you cut it using the same restriction enzymes, the sticky ends will all match up perfectly, and you'll be able to insert it into your bacterial plasmid. So when the sticky ends match up, it now all binds together, and now this is inserted back into a bacteria. So here are the steps that I just mentioned, all right? Step 1, you got to locate the DNA that you want in the human gene. So in this process, we're trying to put human DNA into a bacteria. So you locate the gene that you want, and then you isolate it. Once you've located and isolated it, you cut around it. See how we cut around the gene and release the gene? That's step 2. Step 3 says you got to cut the plasmid open. Well, you got to cut the plasmid open using the same restriction enzyme. Why? If you use the same restriction enzyme, you will have sticky ends that match up perfectly throughout. Okay. Step 4, sticky ends will match up, and you actually can combine them together. You use this enzyme we've talked about in a previous chapter called ligase, and ligase will reattach the pieces. Okay, so now we have a plasmid that is containing human DNA. So it's now recombined or recombinant DNA. You insert it back into a bacteria. I already mentioned to you, how do you insert it in or take it out? You put the bacteria on ice. It creates larger membrane proteins. The protein channels in the membrane get bigger, so things can slide out and things can slide back into the actual bacteria cell. Once you've inserted it back in the bacteria cell, it does transcription, makes mRNA, and then the ribosome reads the mRNA, does translation, and makes the protein product. So now it is expressing itself, producing a new protein product. So this could be an insulin gene from human being inserted into bacteria. Now you're producing a bacteria factory that is making insulin for you. Now, when you see these problems in class, you're going to see them like this. You're going to get plasmid DNA sequence. You're going to get human DNA sequence. We could not connect this into a circle. It's very difficult to read if this sequence was connected into a circle. So I'm showing you this picture here to show you that this sequence really will connect all the way around and make a circle. Your human DNA sequence is right here, right in the middle here. We have our insulin gene, and there it is right there. Okay, so what is it? What is your job? Your job is to choose a restriction enzyme, and I've given you 3 of them down here. Your job is to choose a restriction enzyme that will allow you to remove the insulin gene and insert it into the circular sequence, basically. All right, so I've given you 3 restriction enzymes. One I've called HAL, one I've called GAP, and one I've called AVI. So your whole idea is which restriction enzyme will allow you to produce a recombinant DNA. Well, the first thing you need to do is you need to label all the cutting sites on the plasmid and the human DNA, and it should look something like this. Okay, so I've labeled them all for you right there. HAL cuts here, TAAT makes a stair step, it cuts there, and it cuts here. Okay, look at that, it cuts through the gene. That's not a good thing. If you're cutting through the gene, you're probably going to damage it. So HAL is not a restriction enzyme that you'll probably want to use. GAP, this restriction enzyme cuts here one time, cuts on the human sequence one time. So it's not cutting out the gene. The GAP enzyme doesn't work very well. Okay. AVI, the third enzyme, cuts with this sequence. It's labeled here, here, and there. And AVI cuts the plasmid in one spot, which is perfect because we want to cut it open, and cuts the human DNA in two spots, here and here. And what I've done is I've color-coded these things here, showing you where the cuts are all taking place. So if you notice the red one, which is HAL, cuts through the gene. The blue one only cuts on one side of the gene. The green one, which is called AVI, cuts on both sides of the gene, so it allows you to cut it out, and it cuts the circle in one spot, allowing you to insert it. Now, if you make all of the same cuts, like I am showing you here, all the sticky ends will match up perfect, and it will allow the insulin gene to be able to be inserted in the plasmid sequence. So the correct answer should be AVI is the correct answer. This is the restriction enzyme that will allow you to produce recombinant DNA. It has to be one enzyme, and it has to make a total of 3 cuts. 1 cut on the circular sequence, 2 cuts around the gene that you are trying to insert into the circular sequence. We are done for this lecture.